Well, hello everybody, and welcome to this week's Infection Control Matters. It's Martin Kiernan here, and my special guest today is Professor Brett Mitchell. Brett, I believe you've been on the podcast before.
Hello, Martin. Yes, just a few times, in fact.
Okay, now we're going to have a chat about a paper that Brett has. It's piqued his interest a bit, and it's a paper from the European, it's ECDC Nosocomial Infections Study Group, headed up by Professor Hilary Humphreys and Marguerite Voss and a few other key players. But this is a paper published in the Journal of Hospital Infection. And Brett, you're going to tell us about it.
Yep, thanks, Martin. Yeah, it was published. It's a preprint at the moment. Maybe by the time this podcast goes out, it'll be in full press. But it has been peer reviewed. So in this, this is a survey that the ESCMID Study Group for Nosocomial Infections did across Europe. And, the purpose of the study was to do a bit of a survey about environmental sampling practices of innate hospital surfaces. And, I've always had an interest in, obviously, in cleaning and environmental services, but this sort of concept of swabbing has always been an interesting one because I can see the pros and the cons and the bones of contention with it. So I was really interested when I saw this because I thought, oh, wonderful, it'd be great to see what people are doing about swabbing the environment in their institutions. So the survey was sent out through some member groups, the ESCMID Study Group, the European Society for Clinical Microbiology and Infectious Diseases, Hospital Infections Society, and the Study Group for Nosocomial Infections. And it was sent out in towards the end of the second half of last year in 2021. Now, what they were really interested in looking at were three things. They were interested in the sampling practices that people were doing. They were interested in the role that people had in environmental sampling and the sampling process methods. So three things I think are quite pertinent to when we're thinking about environmental sampling, what we might do. There is a big gap in this paper for my mind. If I look back now, and I know we usually talk about limitations at the end, but I would have loved them to be asking how.
Bit brutal going for limitations
At the beginning. Well, it's not a limitation. It's something I think, I wonder if they thought about doing this, and be fascinated to hear if they thought about it or they just didn't want to go down that route. But how you actually swab, not what you swab with, but what surface area do you do? For example, I didn't see covered in this survey. I stand corrected, but I couldn't find information on that. And I think that would be fascinating, perhaps, for a follow-up piece of work. But anyway, they had 89 people respond to this survey. We're a bit unsure about what the denominator is because that's near impossible when you send out surveys through study groups. That's just a given. But the range of people who responded, clinical microbiologists, infection control practitioners were the vast majority of people who responded, and across 21 different countries. So quite a good representation. That said, just over half were from the Netherlands, and about 20% were from the UK. Maybe because the Netherlands can speak really English exceptionally well, and the UK try, and I'm having a
trying to speak English well.
Speak English well, just like me tonight.
Anyway, yeah, me too.
So look, that was a good cross section of different countries, but predominantly from the Netherlands and the UK. So a few things that in terms of the results, the respondents reported this, and this is an interesting one because later on I might come back to this, but they reported that they had a good to excellent knowledge of sample collection.
Yeah, we're good at thinking we're great, aren't we?
Well, that's right. In fact, let's just go straight to that point, Martin, because I think one of the things that the authors found was that the respondents claimed to have a good to excellent knowledge at the start of the survey, but a substantial proportion of these respondents were not able to answer the relevant questions. So interesting. Self-evaluation is always hard, of course, but it does say something to that type of question. It's always a difficult one to gauge when people self-report knowledge. But similarly, 75% of respondents also reported they always or usually had a sampling protocol. So they had a process by which they were going to go about their sampling, which was good to see. It would have been great to hear what that sampling protocol actually involved. So on those sort of 3 points that they looked at, the first thing they wanted to look at was sample locations. So what sites did people sample? And look, they did vary a lot. UK respondents, interestingly, never sampled the privacy curtain for any microorganism. Now, I'm not sure.
Right.
I'm not sure why that's the case. Quite fascinating.
I wouldn't mind, but Tim Boswell's published loads of papers on sampling privacy curtains and has grown all sorts and has detected the source of outbreaks related to that. That's
right.
in Nottingham.
I remember a C. diff outbreak that I was involved with, and we privacy curtains played a big role in that one. So yeah, but it's interesting. So bearing in mind, quite a few respondents from the UK. So they say they never sampled the privacy curtain. The Dutch respondents never sampled the mattress and the patient locker for Pseudomonas. This is where it starts to get interesting because there are such intricacies with how people were sampling or not sampling different locations. And then on top of that, sampling or not sampling certain locations, but variation of what the organisms were that they were actually sampling for.
Mm.
And there was another little interesting thing. Among the Dutch, UK, and Irish respondents, there was consensus not to sample the shower head, shower drain, or the toilet bowl for MRSA. So, anyway.
Okay.
Look, probably not unexpected for that one. On the sort of CPE front, that was another interesting thing. So dry sites were mainly sampled for CPE, except in the Netherlands, where these were frequently sampled. And wet or damp sites were mainly assessed for CPE in the UK and Ireland, as well as Pseudomonas. So again, it just goes back to that point. There's just this enormous variation in sites and what people were sampling.
Is that a product of the literature? Because there's been that many papers about drains and splashing from drains and, finding bugs down drains that people are actually actively looking down there and thinking that's a problem. And personally, I don't necessarily think it's such an issue because it's how does it get out of the drain I'm more worried about. If you can clear the organisms from the top end so that you don't get a splash, that's an issue. But there's been so many papers again recently, despite the fact that we actually knew this back in the 80s. Is that just bias because that people are seeing these things in the literature and then thinking, oh, maybe I'll go and do that sampling myself?
Maybe. And I can't remember your analogy, Martin, where you talk about sinks being the toilet of the, I can't remember what you say now, but basically what you're getting at is what do you expect to find because you're washing away all the bugs from your hands?
Well, it's the whole point, isn't it? If you go to the toilet and wipe your backside, for those of you sensitive nature, I apologise, then you're going to wash your hands afterwards, hopefully. So therefore, what you've got on your hands when you've been cleaning yourself goes down the sink.
Yeah.
Duh.
That's right. You're going to find it.
So I'd be more worried to not find organisms down the sink, to be absolutely honest. And if staff are looking after a patient and they've been cleaning them up and they've been fecally incontinent, there's a chance they may have fecal matter on their hands, we recommend they wash their hands in the sink with soap and water. If we get C. diff cases, we say use soap and water. We know C. diff spores come out of the rear end. Therefore, those are the sort of things you should find down a sink. So to me, sampling sinks and finding a whole range of multi-resistant organisms tells me far more about people's hand hygiene than it does the 95% compliance on the wall door.
Yeah, good point. So, I wonder, there are different priorities in different countries, and the authors allude to that quite nicely in the discussion. So, countries that might have a specific priority for VRE because, it's either they might have a low prevalence, like the Netherlands might want to maintain a low prevalence of VRE. So they might well have a specific targeted strategy for swabbing VRE in certain situations that might be very different to a country or that has a different epidemiology of that particular bug. So perhaps that's one of the reasons why we see some of the variation in this survey as to what people are sampling and what they're looking for when they actually sample.
I suppose that's reasonable, isn't it? You use your local epidemiology. I mean, I've done sampling of the environment in the past, and funnily enough, I was going through some of my old papers the other day, and I found a photograph of myself from the early 1990s on top of this is in the local paper, on top of a stepladder, wearing a shirt and tie, long sleeves, swabbing the top of a curtain rail for MRSA because we'd had a ward closed because of MRSA, in the days when MRSA was a superbug.
Yeah.
Just a bit embarrassing. And I've done sampling of the environment for VRE. The thing is, your method determines whether you grow the thing or not. And that's also the problem, because I found that we didn't grow anything from a straight swab, but if we put them in a salt broth like we used to do for MRSA screens back in the day, then you would actually grow organisms if you enrich them first. And I think Jean-Yves Maillard and his group came up with why that's the case, because these organisms are out there in dry biofilm, or they're lying around in the environment in a sessile form. So it's not like when you look down a drain, you see all the sludge, and that's clearly going to be a lot of that is biofilm. But in the dry environment, you can't detect biofilm, and that's for me one of the limitations of this paper. It doesn't mention biofilm at all, so nobody's actually looking at the actual method of what you do with the swab to try and detect something.
Yeah, I agree, man, totally. And the next thing they leads on nicely. The next thing in the results are about the sample methods. They talk about Dutch respondents having preference for flock swabs and never using sponges, UK preferring cotton or flock swabs and sponges, and never using, wipes, and no one using dip slides. So that's interesting. The variation in the sampling method by which people are actually using. But I think, as I said at the start, the thing that would be really interesting as well is to know is how people are actually swabbing in terms of the amount of surface area that they're swabbing. How is that relative to cleaning? And what about dry biofilms? And so understanding those are really important, because you see a lot of studies out there that report on environmental contamination as one of the outcomes, for the studies. But
they don't tell you how they did
there's
it.
just too much variation to be able to use it meaningfully between studies.
We sampled the environment, but how did you do it? And I vividly remember chatting to the late and great Tina Bradley from the Hospital Infection Research Laboratory at Birmingham about sampling, and she was a great advocate of using a sponge because she felt you could actually put a bit more pressure on the surface, and therefore you might have more chance of removing organisms that were more embedded onto the surface in a sort of dry biofilm type way, rather than just waving a swab over the top and then what sort of surface area do you do? And also the other issue is your lab accredited to do surface sampling specimens? Because a clinical microbiology lab in a hospital probably isn't, even though my lab were very generous and would always, we'll have a go and, we'll use a method, but they weren't accredited to do that sort of sampling. So that's another slight problem.
Yeah, that's a good point, actually. Yeah, that's right. And the third thing they looked at then was the laboratory processing. And so, what they found was that indirect culture methods were preferred for MRSA, VRE, CPE, and direct culture methods for things like Pseudomonas, Acinetobacter. For MRSA and VRE, selective enrichment broths were preferred as well. So again, I'm not going to go through all of it, but there was a fair bit of variation there, perhaps a little bit more standardization in laboratory processing, which I guess you would expect, compared to perhaps a lot of the variation when it came to the sites and the organisms that were being targeted. So, look, I read this and I went, great. I was so excited when I saw it, and I thought, thank goodness someone's done it. And I'm glad that authors have done it. I really am. And it just left me with a lot more questions.
Yeah, well, that's good research, though, isn't it?
That's good research. And I think the authors make a really good point. And it sounds to me like this is the starting point, really. And it's a great starting point, because I think ultimately, reading between the lines, perhaps what the ultimate goal here is trying to come up with some consensus recommendations, guidelines, whatever you want to call them, to perhaps standardize some of these methods. And if they can achieve that, and this is the starting point for that, all kudos to all the authors on this paper, because that's just so desperately needed. And I'd love to see some kind of standard or guideline or protocol for some of these, where look, it's not saying we can only do this in certain periods. There's lots of reasons why we'd go and do swabbing as part of epidemiological outbreaks and investigations. But when we do it, how are we going to do it? and standardize that. And if we're going to do it in the context of research, then I think we can be thinking about things a bit more in terms of what we swab and some of the other detail that aligns with that.
Yeah, my nervousness is that unless you really think hard about doing your swabbing, you stand a fairly good chance of disproving your theory that environment might be an issue for you because you don't get your technique right. And I'm going to give a shout out to Elaine Cloutman-Green, who, along with Stacey Rawlinson and Lena Sirich, published a paper in the Green Journal, in the Journal of Hospital Infection, I think it was about 2019, which is a review of methods of taking specimens from samples, and I'll put a link to it in the paper. It's the paper's called "How to Carry Out Microbiological Sampling: A Review of the Current Evidence." I think it's a very nice piece of work. It says it's very difficult to actually draw overall conclusions. Most of these studies were based in laboratories and not undertaken in a real hospital environment where lots of different things like the sampling recovery could affect the variables present. But they did make some recommendations, and I think that also is a good starter as well. So that's a nice paper. I know she and Lena and I have got a study grant from the Healthcare Infection Society at the moment. We're working on that sort of piece of work as well. So,
interesting.
And Sam is the lead on that
We'll
one.
have to get some more on that. I'd love to hear more about that at some point, Martin.
Yeah.
Be,
well, we'll get Lena and Elaine and Sam on, I think, because that's it'd be interesting to find out. Because they've done a lot of sampling at Great Ormond Street, and they sample the environment for adenovirus and other viruses, and they're very expert at it. So I think they'd be good people to get on sometime.
It's such a common question that gets asked. So I think that is going to be a topic. We do need to get them on, and I think that's something that people might well be interested in. If it is, please do give us a shout out on Twitter, because we'd love to hear about topics you're interested in as well. So look, so I think this is a great piece of work. It's going to be a starting point for many things to come, hopefully, and combined with some of the work that sounds like going on in other areas. That's probably why it piqued my interest there, Martin. I guess anything in the environment often does, but this one in particular.
Yeah, I like reading a paper that makes me go, oh yeah, but, and have we thought about that, and why haven't we done that? So, and this is one of those. I think it's one that makes your brain work, and I like those sort of papers.
It does indeed. Well, Martin, as usual, great chatting, and thanks to everybody who's joined us for this Infection Control Matters podcast. And as I sort of mentioned, we would love to get your feedback on the podcast and comments on what you want in the future as well. So please do get in contact with us, and Twitter is probably the best way to do that. But
I think it's the only way to get in touch with
us. It's the only way we promote this.
That's true.
All right. Thanks very much, Martin. Great to see you.
Cheers, Brett.
Bye for now.